Hello, my name is Joshua Mugabe. Welcome you to Nucleic Acid. What are nucleic acids? What is the historic origin of this name? DNA and RNA, the nucleic acids, the molecules responsible for hereditary information that commands the protein synthesis in living beings. The name nucleic is derived from the fact that they were discovered within the nucleus by the Swiss biochemist Friedrich Miescher in 1869. At that time, it was not known that those substances contained the hereditary information. Of what units are nucleic acid constituted? What are the chemical entities that compose that unit? Nucleic acid are formed by sequence of nucleotides. Nucleotides are constituted by one molecule of sugar, deoxyribose in DNA, ribose in RNA. These are bound to one molecule of phosphate and to one nitrogen-containing base, and the bases are adenine, uracil, cytosine, and guanine in RNA. Adenine, thymine, cytosine, and guanine in DNA. What are pentoses? What organic groups do pentoses belong? A nucleic acid formed only of one type of pentose? Pentose, these are carbohydrates. They are made up of 5 carbons. Deoxyribose is a pentose that constitutes DNA nucleotides, and ribose is the pentose that is part of RNA nucleotides. In which two groups can the nitrogen-containing base that form DNA and RNA be classified? What is the criterion used for that classification? The nitrogen-containing bases that form DNA and RNA are classified as pyrimidines and purine bases. By the analysis of the structure formula of these nitrogen-containing bases, it is possible to realize the 3 of them: cytosine, thymine, and uracil have only one nitrogenized carbon ring. Others adenine and guanine, they have two nitrogenized associated carbon rings. Concerning the nitrogen-containing base that participate in the nucleotides, what is the difference between DNA and RNA? In DNA, nucleotides can be formed of adenine, thymine, cytosine, or guanine. Whereas in RNA nucleotides, these ones can also contain adenine, cytosine, guanine. However, instead of thymine, there is uracil. Which are the nucleotides portion that bind the formation of nucleic acids? What is meant by 5 and 3 prime extremities of nucleic acid? The phosphate group of one of the nucleotide binds to the pentose and the other nucleotide, so to make the polynucleotide chain. Each extremity of a DNA or RNA chain can be distinguished from the other extremities according to their terminochemical entity. The phosphate-ended extremity is called 5' and the pentose-ended extremity is called 3' extremity. So, DNA or RNA chain can be run along the 5 to 3' way or along the 3' 5 way. These ways are important in several biological functions of DNA and RNA, since some reactions specifically occur following one way or the other way. Bacteria are prokaryotic cells. That is, they do not have a membrane-delimited nucleus. Eukaryotes have cells with delimited nucleus. Where in these type of cells can DNA be found? In eukaryotic cell, DNA is found within the cell nucleus. In prokaryotic cell DNA, it is found in the cytosol, filled space inside the cell. Other DNA molecules can also be found within the mitochondria and chloroplast, specialized organelles of eukaryotic cells. Who were James Watson, Francis Crick, and Maurice Wilkins? Watson from North America, Crick, Britain, Wilkins, New Zealand, were the discoverers of the molecular structure of DNA, the double helix, made of two polynucleotide chain paired by their nitrogen-containing base. They won a Nobel Prize in Medicine in 1962 for the discovery. According to Watson-Crick's model, how many polynucleotide chain does DNA molecule have? DNA molecule is formed of two polynucleotide chain bind in antiparallel mode, 5 to 3' and 3' to 5, and forming a helical structure. What is the rule for the pairing of nitrogen-containing bases in DNA molecule and in the RNA? Is this last question appropriate? The rule of pairing the nitrogen-containing bases of the polynucleotide chain that form the DNA molecule is pyrimidines bases bind to purine bases under the condition that thiamine binds to adenine and cytosine binds to guanine. In RNA, there is no binding between the nitrogen-containing base. That is because RNA is formed of only one polynucleotide chain. Differently, DNA is formed of two chains. It is therefore not correct to question the base pairing in RNA. What is the numeric relation between pyrimidine and purine bases in the DNA molecules? Is that relation varied in RNA molecule? The DNA molecule is made up of two bound polynucleotide chains that form a helical structure, what we call the double helix. The binding of the two chains is between the nitrogen-containing bases, and it is always obeys the following rules. Adenine, a purine base, binds with thiamine, a pyrimidine, and guanine, a purine base, binds with cytosine, a pyrimidine base. Therefore, in one molecule of DNA, there will be the same number of adenine and thiamine, and the same number of cytosine and guanine. The quantities of purine and of pyrimidine bases then will also be the same in a 50% proportion for each type. The relationship between adenine and thiamine, cytosine equals to guanine, or adenine stroke thiamine equal to cytosine stroke guanine, which equals to 1. This is called the Chagaff's relation, and the pairing rule described above is known the Chagaff's rule. In RNA, there are not two nucleotide chains. RNA is a simple chain molecule, and there is no necessary proportionality of the nitrogen base containing bases. Which type of chemical bond maintains the pairing of the chain in DNA molecule? To form DNA molecule, purine bases bind with pyrimidine bases by intermolecular bonds called hydrogen bonds. Hydrogen bonds occur when there is hydrogen near one of the electronegative elements like fluorine, oxygen, or nitrogen. In such conditions, hydrogen looks like having lost electron for those elements, and a very strong polarization is created. The highly positive hydrogen attracts a pair of electrons from the other molecules, making the hydrogen bond. What is the completing sequence of the nitrogen-containing bases for adenine, guanine, cytosine, cytosine, guanine, thymine, thymine, adenine, adenine, cytosine fragments of the DNA chain? It is thymine, cytosine, guanine, guanine, cytosine, adenine, adenine, thymine, thymine, guanine. What is the name of the DNA duplication process? What is the main enzyme that participates in it? The process of copying or duplication of DNA molecule is called replication. The enzyme that participates in the formation of the new DNA chain is the DNA polymerase. There are also other important enzymes in the replication process: the helicase, the gyrase, and the ligase. Why is it not correct to assert that DNA self-replicates? DNA is not completely autonomous in its duplication process because the replication does not occur without enzymatic activity. So it is not entirely correct to assert that DNA self-replicates. How do the two complementary nucleotide chains of DNA facilitate the replication process of the molecule? The fact that DNA molecule is made up of two polynucleotide chains whose nitrogen-containing bases form hydrogen bonds facilitates the duplication of the molecule. During DNA replication, the binding of the two chains is broken, and each of them serves as a template for the formation of the new nucleotide sequence along it, with the help of the enzyme DNA polymerase, obeying the pairing rule adenine with thymine, cytosine with guanine. At the end of the process, two double helix of DNA are produced, each made of an original template chain and a new synthesized polynucleotide chain. What are the chemical bonds of the DNA molecule that are broken for replication process to occur? During DNA replication process, hydrogen bonds between the nitrogen-containing bases of the polynucleotide chains are broken. As a result of DNA replication, two DNA molecules come into existence. Why is it not correct to assert that two new DNA molecules are created? What is the name given to the process of concerning that fact? During replication, each chain of DNA molecule acts as a pairing new nucleotides. After the process, two new reformed chains made with the union of these nucleotides appear. Then two DNA molecules are created, each with one chain from the original molecule and one new chain formed by the new nucleotides. Thus, it's not entirely correct to assert that replication produces two new molecules of DNA. It is better to affirm that two new half molecules are created. For this phenomenon, DNA replication is called semi-conservative replication. Does DNA replication occur in cell division? The answer is yes. DNA replication occurs in mitosis as well as in meiosis. One characteristic of DNA molecule is its replication capability. What are the consequences of failure during DNA replication? Ideally, DNA molecules should replicate in a perfect way. Sometimes, however, failures of the duplication occur with the alternation, that it might be deletion, addition, or substitution of one or more nucleotides in the molecule. Those mistakes or mutations therefore makes the change in the protein synthesis process too. For example, the production of important protein for cells or tissues may be suppressed, new utils or inductile proteins can be created, and many others. The mistake of DNA replication results in the alternate genetic material are some of the main creative forces for biological evolution and the diversity of the species. Mistakes may happen during the very copying process. The same is true for DNA replication. Are there correction systems in the cell that try to fix these mistakes? Under which situation are the mistakes carried only by the individual owner of the cell within which the mistake has occurred? And in which situation are they transmitted to other individuals? The cell is equipped with an enzymatic system that tries to make or fix the mistake of DNA replication process. This system, however, is not completely efficient. DNA replication mistakes are kept in the regional individual where failure occurred when the phenomenon affects somatic cells. If replication mistakes occur in the formation of the germline cells, those are the gametes, DNA alteration may be transmitted to the offspring of the individual. Where can RNA be found within the cell? In the eukaryotic cell, nucleus RNA can be found dispersed in the nuclear fluid along with the DNA and as many constituents of the nucleolus. In cytosol, that is in eukaryotes and in bacteria, RNA molecules can be found free as structure of constituent of ribosomes. Organelles specialized in protein synthesis or even associated with them in the process of making proteins. Mitochondria and chloroplasts also have their own DNA and RNA. Does RNA molecule have two polynucleotide chains like DNA? Only DNA has two polynucleotide chains. RNA is formed by just one polynucleotide chain. What is the production of RNA called? And what is the enzyme that catalyzes the process? The making of RNA from information contained in DNA is called transcription. The enzyme that catalyzes the process is called the RNA polymerase. What are similarities and differences between transcription process and replication process? A DNA polynucleotide chain serves as a template in the replication, DNA duplication, as well as in transcription, RNA formation. In both processes, the pairing of the two nucleotide chains of the original RNA molecule is broken by the breaking of hydrogen bonds for the chain to be exposed as a template. The reaction is catalyzed by specific enzymes in transcription and in replication. In replication, the enzyme DNA polymerase catalyzes the formation of the new polynucleotide chain using free nucleotide in solution and putting them in the new chain according to the DNA template exposed and to the rule. Adenine goes with thymine, cytosine goes with guanine. In transcription, the enzyme RNA polymerase makes new polynucleotide chain according to the DNA template exposed, obeying, however, the rule of adenine with uracil, cytosine with guanine. In replication, the original template of DNA molecule is kept bound by hydrogen bonds to the newly formed DNA chain, and the new DNA molecule is then created. In transcription, the association between the template DNA and the newly formed RNA is undone, and RNA constitutes of only one polynucleotide chain is liberated. What are the three main types of RNA? What is meant by heterogeneous RNA? Messenger RNA, MAO, transfer RNA, tRNA, and ribosome RNA, rRNA, are the three main types of RNA. The newly formed RNA molecule, a precursor of messenger RNA, is called heterogeneous RNA. The heterogeneous RNA bears portions called introns and portions called exons. The heterogeneous RNA is processed in many chemical steps. Introns are removed and messenger RNA is created, formed by only of exons, the biological activity nucleotide sequence. Concerning their biological function, what is the difference between DNA and RNA? DNA is a source of information for RNA production, that is transcription, and thus protein synthesis. DNA is still the basis of heredity due to its replication capacity. The messenger RNA is a template for protein synthesis. Translation. In the process, transfer RNA and rRNA also participates, since the first carrier amino acid for the polypeptide chain formation and the second is a structure constituent of the ribosomes, the organelles where proteins are made. Is there any situation in which DNA is made based on RNA template? What is the enzyme involved? The process in which DNA is synthesized having as a template of RNA chain is called reverse transcription. In cells infected with retroviruses, RNA viruses like the AIDS and SARS viruses, reverse transcription and the DNA is made from the information contained in the viral RNA. Viral RNA within the host cell produces DNA with the help of the enzyme called reverse transcriptase. Based on DNA, the host cells then makes the viral proteins. New viruses are assembled and viral replication occurs. Do phosphates and a pentose group give homogeneity or heterogeneity to the nucleic acid chain? What about the nitrogen containing groups? Supported by that, which of those groups is equipped to directly participate in the highly diverse and heterogeneous genetic coding, i.e., which of those groups is the basis of information for protein production? The phosphate and the pentose group are the same in every nucleotide that form nucleic acid, and so they give homogeneity to the molecule. The nitrogen-containing bases, however, can vary among adenine, thymine, cytosine, guanine in DNA, and uracil in RNA. This variation provides the heterogeneity of the nucleic acid molecule. Homogeneous portion of the molecule solidly would store any information by the same reason that a sequence of the same letter of the planet cannot make many words with different meanings. The nitrogen-containing bases, on the other hand, because they are different for different types of RNA or DNA, can make different sequences and combinations that allow the density of the genetic code. Thank you very much for participating in nucleic acid. Please do like, share, and subscribe.